In this video, we are going to talk about animal viruses and their replication cycle. So there's going to be a lot of similarities between what we talk about now and what we just talked about in the previous video where we discussed bacteriophage replication cycles. Now, the big difference here is that animal viruses infect animals, right? Eukaryotic cells. So kind of the location of where things happen is going to be a little bit different because of the organelles within animal cells. But the overall gist is going to be very similar, okay? So I have the steps of the animal virus replication cycle typed out here. Notice there's a lot that's similar. They're going to attach to the host cell. Now they're going to have to get into the host cell, okay? And eventually they're going to make more viral nucleic acid and viral proteins, that biosynthesis step. Eventually they're going to spontaneously assemble in maturation, and eventually they're going to exit the host cell. So there's a lot of similarities here. But notice steps 2 and 3 are a little bit different. Okay, that's because animal viruses are just a bit more structurally diverse and a little bit more complex. Now, with animal viruses, this is where we're going to see some viruses that have envelopes. Remember, that's where they've taken part of the host cell plasma membrane. And some animal viruses are what we call naked viruses. They do not have an envelope. So no matter what, as you see in this picture, we're going to talk about influenza virus, one most of us have experienced the wrath of before. So here the virus is attaching, so it's outside of the host. Remember, the spike proteins are going to be attaching to host receptors. Now the virus is going to enter the cell. And there's two different ways this can happen. As you see in this picture, sometimes the entire cell enters into the virus. I'm sorry, into the host, okay? So we see the intact virus in here. That's called endocytosis, when the whole thing just gets taken into the cell. An alternative entry tactic is called fusion, where the envelope might fuse with the plasma membrane because they're both host plasma membrane, and it just kind of dumps inside of the host the genetic material protected by the capsid, okay? But no matter if it's from fusion or endocytosis, the genetic material is still protected, whether it be just by the capsid or by the envelope and everything else, okay? So that's why there's this unique step, this uncoding step, because we need, well, not we, the virus needs to release the genetic material into the host cell. That's the only way the genetic material is going to be able to be copied. And that's the only way the genetic material is going to be used to go through transcription and translation to make more viral protein, right? In order to make lots of other viruses, more genetic material has to be made, and more capsid protein and all that sort of stuff has to be made as well. The only way that can happen is if the viral genetic material is exposed. So that process is called uncoding. You can think of uncoating as the virus falling apart. It disassembles so as to release that genetic material into the cell so it can start to take over the cell. And so that's what's happening here, okay? And so in this case, the genetic material is heading into the nucleus, so we can probably guess, well, because that's where just certain things are going to happen. I'm going to skip all the unnecessary nitty-gritty details. So once it goes to where it needs to go, it is going to start making more copies of the genetic material. It's also going to start doing like translation and things like that to make more viral proteins. So that's biosynthesis. Then it's going to start to spontaneously assemble. So it's kind of just showing you the initial stages of that here. That's me maturation. And then once the intact virus particles are all assembled, you can see this one is pushing its way out. Okay, now remember, some viruses are going to push their way out and steal some of that host plasma membrane, a process called budding. That's what our enveloped viruses are going to do. But viruses that aren't trying to steal the plasma membrane, they tend to just literally punch holes in the host cell and leave by lysis, much like we saw with the bacteriophage. Okay, so again, the virus is going to attach to the host cell. It's going to get into the host cell, either by endocytosis or fusion. Then the virus is going to fall apart in order to release or expose its genetic material. That process is called uncoating. Once that genetic material is exposed, it's going to start doing, depending on if it's DNA or RNA, it's going to start doing, DNA replication, transcription, translation. It's making lots of copies of that genetic material. It's going to make lots of copies of the capsid proteins and the spike proteins and all of that. They're going to spontaneously assemble into intact virions. And from there, it's going to exit the host, either by budding, where it's going to steal part of the plasma membrane and have an envelope, or by lysis, which is what our naked viruses do. All right, so a quick note about RNA viruses. Because RNA viruses, they can't just use our RNA polymerase, because RNA polymerase is used to reading DNA, right? That's how our RNA polymerases work. We start with DNA and we make RNA. Well, an RNA virus doesn't have any DNA, so they can't just steal our RNA polymerase. It's not going to work. So RNA viruses bring their own type of RNA polymerase, but notice it's an RNA-dependent RNA polymerase. And what that means is this RNA polymerase starts with a molecule of RNA and it creates an anti-parallel complementary RNA molecule, right? This is something our cells do not have. Our cells cannot do this, right? So it's an added layer of complexity for these RNA viruses. And so we actually describe the RNA that a virus has as either being positive sense or anti-sense. A positive sense of RNA runs 5' to 3'. So it acts a lot like messenger RNA. It can use our ribosomes, it can use our transfer RNA, it can immediately undergo translation and start making proteins, right? Because our cells know what to do with 5' to 3' RNA. However, because of this RNA-dependent RNA polymerase, if the genetic material that's exposed is a 5' to 3' RNA, then the polymerase here is going to make an anti-parallel and complementary strand of RNA. Well, that is what we call antisense, because that piece of RNA runs 3' to 5'. Our cells have no idea what to do with this. Our host cell machinery does not ever get exposed to antisense RNA, okay? So what's going to happen is that same polymerase is going to make another copy of RNA. That way, it's going to make an anti-parallel, which is 5' to 3', complementary piece of RNA. Okay, so it's essentially doing its own RNA replication. Okay, now I know y'all, this is kind of confusing. It was confusing to me in graduate school. I'm not going to test you on all the details of what's, how many times RNA polymerase has to do all the things. But I just want you to understand that RNA viruses cannot use our RNA polymerase. It has to bring its own, because it has to have a way of copying its RNA genetic material, because all the new viruses that are made are going to need copies of this RNA, and our cells don't have a way of doing that. So the virus brings its own polymerase that's going to copy the RNA for it. And then to kind of wrap up this conversation, I want to show you just how complex some viruses are, like retroviruses, which HIV is a retrovirus. Retroviruses take complexity to a whole nother level. Okay, these are RNA viruses, but you may have heard that somebody that gets infected with HIV, it stays with them for life. Okay, and it does. And that's because the viral genetic material integrates into the host genetic material. Now, you might be thinking, wait, Chelsea, if HIV is an RNA virus, how is it going to integrate into DNA, right? They're not the same thing. This is why retroviruses are so complex. Retroviruses bring with them their own enzyme called reverse transcriptase, which you can see here on this second bullet. Now look at this, look at these two words. Do we see transcriptase? Doesn't that sound like transcription? So for us, transcription is DNA and we make RNA. Well, what is the reverse of that? Starting with RNA and making DNA? Exactly. So the HIV comes with RNA. It uses this viral reverse transcriptase to create viral DNA. Okay, so that's what's happening over here in this picture as well. So the viral DNA is made courtesy of reverse transcriptase, and now it can be integrated into the host DNA because it has a second viral enzyme called integrase, which integrates the viral DNA into the host DNA. And you can see that right here. And when the viral DNA is incorporated into ours, it's called a provirus. Okay? And actually, if you go back to the bacteriophage replication cycles, when the bacteriophage DNA incorporated into the host DNA, that was called a prophage. Because remember, we use phage kind of instead of the word virus when we're talking about ones that infect bacteria. So prophage happens in the lysogenic replication cycle for some bacteriophage. Provirus forms when an animal virus DNA integrates into the host DNA. And so this is how the virus is with somebody for life, okay? But, oops, this virus, when things get stressful, it can take over our transcription and translation processes, make new viruses, and then that's when outbreaks occur. This is similar for herpes viruses as well. Now, herpes is not a retrovirus, so it's not quite this complex, but herpes is with somebody for life because the herpes virus DNA gets integrated into the host viral DNA. So it also forms a provirus. All right, so I want you to take what we've just talked about animal virus replication, about how RNA viruses are special because they have to have their own unique enzymes in order to replicate. Think about how retroviruses are complex and different than other viruses. And I want you to look at this meme. And this was posted actually by a family member of mine back in the beginning of 2021 when the COVID vaccine was coming out. And this is not a political thing. This is science, okay? I am never going to get political in this class. That's not what this class is about. But I want you to think, what does the scientific community know about viruses, okay, that make this meme really misleading, okay? Because when I first read it, like not even trying to think critically, and I was like, this sounds like a good point. Once you think about all the things we've talked about, it's not near as simple, right, as it seems. That's when I had to be like, Chelsea, stop. You have a degree in this. Let's actually think about this. So here's what it says. 40 years worth of research for HIV, no vaccine. At least 100 years of research for cancer, no vaccine. Ongoing research for a common cold, no vaccine. Less than a year for COVID-19 vaccine, all past that shot, wake up people. So I saw this a lot, like I said, on social media. And I want you to pause this for a second and think about it. What would be a scientific response to this meme? I'm not asking whether or not you think people should get the vaccine or any other conspiracy theories or anything like that. Think about what we've talked about the diversity of viruses, and think about what a good response to this would be. Then unpause this, and I'll give you just a little bit of what I would say, okay? Because I've actually had to have this conversation several times with people. I'm gonna keep it simple because this video is already getting pretty long. So for one thing, we've just seen how different HIV is from other viruses. We don't see other viruses having reverse transcriptase, for instance, where it starts as an RNA virus and makes DNA, right? So comparing HIV to any other virus is like comparing an ant to a whale. Yes, they're both animals, but they are very different animals. They operate in very different ways, right? So it's taken a long time for us to understand HIV and identify these enzymes, identify these processes, okay? It integrates into the host DNA, all right? So there's this whole level of complexity that it's going to be really hard to find a vaccine for it. In only 40 years of research, we've learned a lot, okay? So just give the scientific community some time. Now, 100 years of research for cancer. Cancer is not necessarily caused by a virus, okay? Now, there have been correlations between the human papillomavirus being linked to some types of cancer, okay? But not all cancer is caused by a virus. So many things cause cancer. So there's not even really necessarily a vaccine for cancer in general, right? We do have vaccines against the human papillomavirus, but a lot of cancer isn't caused by a virus or any other like bacteria or pathogen. So vaccines aren't necessarily even applicable here, okay? The common cold, as you're going to learn in Unit 4, is actually caused by tons of different viruses. The common cold is just a general term for those common symptoms, right? Runny nose, things like that. So rhinoviruses cause the cold, adenoviruses cause the cold, coronaviruses, not COVID-19, but other coronaviruses cause what we call the common cold because it's just general vague symptoms. So we're never going to have a vaccine because there's hundreds of things that cause these symptoms. And these symptoms aren't really life-threatening. So it would kind of be a waste of money, time, energy, right, resources to try to tackle the common cold when so many things cause it. Now, in terms of the COVID-19 vaccine, and we'll talk more about this in our vaccine section, but coronaviruses have been around for a long time. We know they typically cause the cold, very general, vague, non-life-threatening symptoms. So the scientific community has been studying coronaviruses for a long time. So when SARS-CoV-2 came out, yes, it was a new strain of coronavirus, but it wasn't like it was the first time we had ever learned that a coronavirus exists. We'd been studying them and their structures, which is what you need for a vaccine, for quite some time, okay? So it wasn't like we just discovered this virus and just made a vaccine for it all in 1 year, okay? There'd been a lot of things already going on. Now we had to tweak things and, you know, tweak our understanding for SARS-CoV-2 because obviously it doesn't give us the symptoms of just a common cold for everybody. So the fact that it was rapidly produced doesn't mean it's unsafe. We had a lot of knowledge that just the general society didn't know about because there was really no need to. So that's what I want to mention. This meme is misleading because viruses are so diverse that just because we have a vaccine for one doesn't mean we should automatically have a vaccine for other. There's a lot of things that have to be considered and known about a virus when making a vaccine. All right, y'all, we're going to go ahead and stop there. Let me know if you have any questions.